Okay, so now let's get into genetic mutations. Okay, so think of the chromosomes, think of the DNA that's making up the chromosomes. And we'll come to the first one, a single gene mutation, meaning a segment of DNA, which makes up a gene, it has mutated, is not normal. Okay, and it's going to impact a trait. Okay, so a single gene mutation is now going to impact a particular trait. This is not the most common genetic mutation. Most genetic mutations are actually multifactorial. There's many genes influencing one particular trait. But we're going to come to diseases that have single gene mutations. And the gene mutation could be on one of the autosomal chromosomes. So those 22 autosomal chromosomes, one of them could have a single gene mutation, or it could be on a sex chromosome. Okay, so typically when we're talking about a sex-linked gene mutation, it's almost always the X chromosome. Okay, we're not going to get into Y chromosome single gene mutations at all. It's typically the X chromosome that we're going to talk about. Okay, so that's just one. So that's one way of having a genetic mutation. Just a single gene has been affected, meaning a single allele has been affected. Okay, what about chromosomal disorders? Number two. So damaged chromosomes, uh, extra chromosomes, or missing a chromosome. So instead of having, say, two chromosomes, you have three chromosomes at a particular site. An example will be trisomy 21. So at the 21st site, you have three chromosomes instead of two. Okay, well, that's Down syndrome. Okay, so what else can happen here? So why could we possibly get a missing whole chromosome or an extra whole chromosome? Well, during meiosis, right, if you go back up to that slide, the chromosomes didn't separate properly, and one cell got a full complement of chromosomes. And now when that cell combines with the other gamete, it becomes trisomy. There's three chromosomes at that site. Now, what about the cell that now didn't get the chromosome, right? So during meiosis, say during division, one cell took both chromosomes and the other cell was left with the missing chromosome. Well, now when it combines with the other gamete, you're now actually missing an entire chromosome. An example of that would be a condition called Turner syndrome, where on the sex chromosomes you're actually XO. So a female is XX, a male is XY. XO is saying, well, you have one X chromosome, but the other one is completely missing. So we call it XO. Okay? So, and that is a complete loss of one chromosome. Okay? Multifactorial gene disorders. So many different genes are involved in most traits. Okay, that's the most common way that you get a trait, your phenotype. So let's say the shape of your knees, or the shape of your ankles, or the shape of your feet. It's multifactorial. So you can have multifactorial gene disorders, many genes involved in a trait. And if those genes are affected or disordered, you can develop certain conditions. And an example would be, say, a cleft lip or a cleft palate, or spina bifida in your back. Those are from multifactorial gene disorders, not a single gene mutation, and also not a missing entire chromosome or an additional entire chromosome. Okay? So I'm just going to show you here what can actually produce a mutation. And let's say it's just a single gene that's involved in this mutation. Well, here's what's happening at the DNA level. So adenine has been replaced through a mutation by thymine. Okay, so since that nucleotide has been replaced, so you were supposed to get an adenine there that would have coded for the for a different amino acid. You now have thymine there that codes for a totally different amino acid in in the production of the protein. So, and this is what happens in sickle cell disease, right? So it's one amino acid that has been substituted, and the protein has been altered because the amino acid is different, which changes the shape and the functioning of red blood cells. And we'll talk about that later. Okay, so this is just an example of a substitution mutation. Okay, so let's look at single gene mutations. Okay, and on the left at the top there is your father, and on the right is your mother. And we're looking at a single gene mutation on whichever chromosome. Okay, we won't choose a chromosome right now. And so what can the offspring get? Okay, well, say that the mother is heterozygous for an allele. So she has a big A and a small a. And the father's heterozygous. So he has a big A and a small a of that allele as well. Okay? What can the offspring get? Well, remember, the offspring gets one allele from the mother and one allele from the father. So you could have two capital A's, or two of you could have a capital A and a small a, and one of you will have a small a and a small a. Okay. What if the genetic mutation was a dominant genetic mutation, meaning all you needed was one capital A and you have the disease? Well, that means 75% of offspring will have the disease because 3 out of 4 have a capital A. What if it's a recessive disease, meaning you need the abnormal gene from both your mother and your father? So you need a small a and a small a. Well, that's only 1 out of 4 offspring. That's 25% will have the disease. Okay, so we're going to start thinking about it this way. And in the top left there, that's called a Punnett square. Draw these out. It makes life so much easier. So at the top is the mother. On the left side, at the top is the father. At the left side is the mother. Okay, she's big A, small a. The father happens to be big A, small a. Okay, what can the offspring do? It helps you not make a mistake when you're trying to calculate what will happen with the offspring. Okay, this is just a reminder. You have 2 alleles at each gene location or loci. Okay, so if they're identical, so you got a big P and a big P from your mother and father, you're homozygous for a dominant allele, since we're saying a big P is dominant. Okay, and we're using a small letter as a recessive. Okay, so what if you got a small a and a small a at a different site, a different gene location? So different alleles, and they're both recessive. Well, you're homozygous recessive. Okay. What if you have a dominant allele and a recessive allele? So one from your mother, one from your father. Well, then you're heterozygous. Okay, they're different. They're heterozygous. Okay, so you can be homozygous dominant, or you can be homozygous recessive. Okay, so we're still talking about gene mutations, single gene mutations. And when we talk about a single gene mutation on any of the autosomal chromosomes, we're going to use the term autosomal. Okay, so on those 22 pairs of chromosomes, if you have a mutation that is dominant, then you only need one abnormal allele to have it express itself as the phenotype. Okay, let me give you an example. Huntington's disease. It's autosomal dominant, meaning if you get the allele or the gene loci from your say your father for Huntington's disease, and your mother doesn't have Huntington's disease, well, you're still going to acquire the disease because Huntington's disease is autosomal dominant. It just needed one allele, and if you happen to get that allele, you're going to express the disease. Okay? So if you were to do a Punnett square, and we'll do some, if one of your parents has an autosomal dominant mutation, then you have, as an offspring, you have a 50% chance of getting the disease. Okay? What about autosomal recessive? Again, we're just talking about the autosomal gene chromosomes. You need both alleles from your parents for you to get the disease. Let me give you an example. Cystic fibrosis. So say your mother doesn't have cystic fibrosis, but she's a carrier for cystic fibrosis. So she's, say, big C, little c. And your father doesn't have cystic fibrosis, but he's a carrier for cystic fibrosis. He's big C, little c. In order to get cystic fibrosis, you need two little c's. You need both recessive alleles. You have a 1 in 4 chance of getting both recessive alleles from your parents. Draw it in a Punnett square. It'll help, okay? All right, so autosomal dominant mutations, if one parent has the autosomal dominant mutation, the offspring has a 50% chance of acquiring it. Autosomal recessive, if one parent has the autosomal recessive mutation, well, nobody's going to get the disease. If both parents are a carrier state, meaning they have just one allele for the disease, it's a 1 in 25 chance, or sorry, a 1 in 4 chance, or a 25% chance of getting the disease. Okay, so we're still on single gene mutations, but we're going to talk about single gene mutations on the sex-linked chromosomes. So that's the X chromosome and the Y chromosome. But really, we're only going to talk about X-linked mutations. Y-linked mutations just aren't that common, okay? So what is this? This is a single gene defect on the X chromosome. It might be coming from your father because your father has an X chromosome, or it might be coming from your mother because your mother has 2 X chromosomes. Okay? So the mutation on the X chromosome will cause a particular phenotype if it occurs in males. And why is that? Because males only have one X chromosome, right? So where do males get their X chromosome from? They get it from their mother, always, because they have to get the Y chromosome from their father. So if there's a defect on the X chromosome in males, it's going to be expressed, since they don't have a protective X chromosome from their father. They only have the Y chromosome. Okay. So if a mutation is recessive, meaning you need both alleles to have the disease, then the mother is going to be a carrier. She won't have any disease. So if one of the mother's X chromosomes is defective, that's not a problem for her. She won't have any abnormal phenotype because she has a normal X chromosome. Okay? That's not the case with the father. If the father has an abnormal X chromosome, he will have an abnormal phenotype because he didn't have another X chromosome to hopefully compensate with a normal allele. Okay? If the X-linked mutation is dominant, well, the mother's definitely going to have it because it doesn't really matter what's on her other X chromosome. The mutation on one of her X chromosomes is dominant. So the allele is dominant, and you only need one dominant allele to express the mutation as the phenotype. Okay, so let's look at what happens with all male offspring of, say, a mother that has an X-linked disease. Okay, well, the mother always gives her X chromosome to her children. To her males, to her sons. Let me try again. So the sons are XY. So all the sons will have her abnormal X chromosome. So the son gets the X chromosome from the mother. A female offspring, or a daughter, will need two abnormal X chromosomes, one from the mother and one from the father, if a recessive condition is going to be expressed. Okay? So let me give you an example. Hemophilia. Okay, so hemophilia is a clotting disorder where patients bleed a lot because they're missing a particular clotting factor. Well, it's X-linked. So who's going to get hemophilia? Because it's an X-linked single gene mutation. So if the mother has the mutation on one of her X chromosomes, she's a carrier, but she doesn't have hemophilia because she has a normal X chromosome. Who's she going to give it to? She's going to give it to some of her sons. So she could give her abnormal X chromosomes to her son, or she could give her normal X chromosome to her son. So we're also going to use a Punnett square for X-linked mutations as well. Okay. How would a female get hemophilia? That's going to be really, really hard because the mother would have to have an X chromosome that's defective and potentially can lead to hemophilia. But the father would have to have hemophilia. He would have to have the abnormal X chromosome. Okay? So the X chromosome coming from the father has to be defective, and the X chromosome coming from the mother has to be defective in order to have hemophilia in a female, since it's X-linked recessive. Okay? This will make more sense when we do Punnett squares. Okay, so let's look at a Punnett square, which really is used for single gene mutations, right? So mutations that are occurring at one site on a particular chromosome. And then we can put it into a Punnett square. And this is going to allow you to predict the genotypes of the offspring of these two parents. It's also going to allow you to predict the phenotype based on what the offspring's genotype is. Okay, so let's look at this Punnett square. So let's just say that in order to develop the disease, you just need one small. You need both small d's. So it's a recessive disease. You need both alleles to get it. Okay. The mother has a big D and a small d. The father has a big D and a small d allele as well. Okay. So capital D, nothing. Capital D, small d, recessive state. Carrier state. You have another capital D, small d, carrier state. They don't have the disease because in order to have the disease, you needed both. Small d, that's only 1 in 4 offspring. That's a 25% chance of getting the disease. If your mother is a carrier and your father is a carrier of an autosomal recessive disease, there's a 25% chance of getting it. Okay, 1 in 4. Okay, let's look at a disorder where if you are, it's dominant, meaning, let's say you just need one big D to get the disease. Okay, so who's going to get the disease now of those 4 offspring if it's a dominant disease requiring only one allele? You have 3 out of 4 children that are going to have the disease because 3 out of 4 children have a capital D, and it's a dominant mutation, meaning you just need one allele. Okay, so 75% will have the disease. Okay, so let's look at brown eyes and blue eyes. Okay, so the father in this case has capital B, and we know that brown eyes are a dominant trait. You just need one allele. Well, this father has two alleles for brown eyes. The mother has blue eyes. Okay, so blue eyes are recessive. So in order to have blue eyes, she has small b, as her alleles. Okay, is anyone going to have blue eyes when you put out the Punnett square there? No, 100% of the offspring are going to have brown eyes. How many are heterozygous for brown eyes, meaning in their genotype, they're heterozygous? Well, all of them are heterozygous, big B, small b, but they still have brown eyes. They all have brown eyes, okay? So none of the offspring will have blue eyes, okay? Now, what if one of those children that has a big B, small b, marries someone that is also big B, small b? Okay, so now they both have brown eyes, by the way, because they have big B, small b. Okay, so now the children are, if you did a Punnett square, you could have 1 in 4 children with blue eyes. Because if you got the small b from your mother and the small b from your father, that child will have blue eyes. Okay, in this case, nobody can get a small b and a small b because the father has 2 dominant alleles for brown eyes. Okay? Okay, so let's just look at this gene mutation. And let's just say, for example, just as an example though, it's for being tall. Okay, being tall is not really a single gene mutation, but this is just to just give an example here so you can understand the inheritance patterns. Okay, in order to be tall, let's say that it is a dominant trait. So you just need one allele. You just need one dominant allele and you're tall. Okay, so we'll call that capital T. And small t is the recessive allele for being short. Okay, let's look at both parents in the Punnett square here that we've made. Well, both parents are, first of all, heterozygous, right? They have a big T and a small t. They're heterozygous. Are they tall? Absolutely, because we already said that you just needed one big T to be tall because we've made the allele a dominant trait. Okay? All right. So both are tall even though they're heterozygous. Okay, what are the children going to be? See, this is where the Punnett square really helps. So one child is going to be capital T, capital T. He's homozygous dominant and he's tall. Okay, so if I asked you, what percentage of people have a genotype that are homozygous dominant for this trait? 1 in 4, 25%. Okay, how many children are going to be heterozygous dominant? So they're still going to be tall, but they have a big T and a little t. Well, that is going to be 50% of all the offspring. Okay, how many children are going to be homozygous recessive and short? Well, you need small t, small t. That's only 1 in 4 or 25%. What if you were asked what percentage of the offspring are tall? Well, you just need one big T to be tall because it's a dominant trait in this example. So 75% of the offspring will be tall. What percentage of children will be short? Well, only 1 in 4 because you needed 2 recessive traits, small t, small t. Okay, so let's do some more examples. And if you feel you totally understand the different types of examples of autosomal recessive, autosomal dominant, then I would just suggest you skip this slide. If not, let's do another one. So autosomal recessive. So again, cystic fibrosis is a perfect example of an autosomal recessive condition. What else is autosomal recessive? Meaning you need both alleles from your parents to be affected with the gene mutation. So sickle cell is an autosomal recessive. Another condition called Tay-Sachs is an autosomal recessive condition. So we're gonna come across some a bit later. This inheritance pattern. Okay, so let's look at the children here, where if you have both blue colors from your mother and father, then you're affected. Okay, so the one on the left, the child on the left, has the autosomal recessive disease. The two in the middle are carriers. They only have one allele that's affected. And the one on the right is completely not, they don't have the disease and they're not a carrier. Okay, completely unaffected. Let's go to the right, autosomal dominant. Okay, so if you're autosomal dominant, you just need one allele to have the condition, right? To express the disease. So what would be an example of an autosomal dominant disease? Well, how about Marfan syndrome, or Huntington's chorea, or polycystic kidney disease? Those are examples of autosomal dominant inheritance. Okay, so let's look at this example on the right. So the father has the affected allele. Okay, his other allele, which is marked as kind of this beige color, is the normal allele. But it doesn't matter because it's autosomal dominant. He has the disease. Okay, so the mother completely unaffected. She has two normal alleles. Okay, at that gene site. Okay, what about the children? All they need is one affected allele to have the disease. So the first child on the left is affected. The two middle children are completely unaffected because they got a normal allele from their father and a normal allele from their mother. And the last child is affected because every time you have a child, the child is going to get one of your two alleles. So it's a 50-50 chance that the father gave the affected allele to any particular child, right? So what percentage of the children have the allele or have the disease? 50%. What percentage of the children are carriers? 0. You can't be a carrier of an autosomal dominant disease. You either have the disease or you have completely normal alleles, right? Because it's dominant. So if you have the allele, you have the gene mutation and it's dominant, well, you have the disease. You can't be a carrier. Okay, and remember, as you practice these with Punnett squares, it is really hard to get them wrong if you get to a level where you're comfortable doing the Punnett square. Okay? So let's go to X-linked diseases. Remember, we're not going to talk about wide-linked diseases. They're just not that common. Most of the ones that we have through history and in the population, they're going to be X-linked, not Y-linked. Okay, X-linked recessive, meaning in order to have the disease, you need two abnormal X chromosomes. Okay, let's look at the one on the left here. So the father is completely unaffected. He only has one X chromosome, and at that site, at that gene site, it's normal. The mother doesn't have the disease, but she's a carrier. She has two X chromosomes. What are going to happen to the children? Okay, so if we look on the left there at the children, okay, the boy has to get, or sorry, the son has to get the Y chromosome from the father. So in this example, he got the normal chromosome or the normal allele from the mother. He's unaffected. Okay, the second girl, she got a normal X chromosome from her dad, and she got a normal X chromosome from her mother. She's completely unaffected. Okay, the third girl, she got a normal X chromosome from her dad, and she got an abnormal X chromosome from her mother. She's a carrier. So she has a normal X chromosome, and she has the recessive abnormal X chromosome, the abnormal allele, but she's just a carrier. She doesn't have the disease. And finally, an affected son. Why? Because he got an X chromosome from his father, and he got the he got the Y chromosome from his father, he got the abnormal X chromosome from his mother. He has the disease. Okay? So you have a 1 in 4 chance of inheriting an X-linked recessive disorder if your mother's a carrier. What's the chance if you're a boy or a son? It's a 50% chance. So if you're a son already, well, you have a 1 in 2 chance of getting that abnormal X chromosome from your mother. So 50% of the boys will have the disease. Okay? And this is how hemophilia goes through the genetics of a family, right? A family tree. So it runs through, the disease runs through the boys. The girls get a carrier state, and when they have sons, they give it to their sons. Okay? Let's look at X-linked dominant, just for another example. Okay. So the dad has a certain disease that's on his X chromosome. Okay. The mother has completely normal X chromosomes. Remember, this is a dominant. If you see dominant, you just need one allele. Okay, let's look at the children. The first one, where did the X chromosome come from in the son? Came from the mother, normal. Okay, what about the girl, the second one? Well, she got an X chromosome from her dad. She has the disease. She just needed one. The son, the third child is a son, totally unaffected because he got his X chromosome from the mother, who doesn't have either alleles affected. And finally, the fourth child, a daughter, she has the disease. So notice how it changes. An X-linked dominant disease that the father had now is affecting the girls. And it's not affecting the boys because the father is only giving Y chromosomes to his sons.